All right, team, good morning or afternoon or evening, whenever you're watching this. This is Miss O'Brien, and we're going to go through and talk about the stages of meiosis. I'm going to be working on paper, but you have access to a digital copy on this in your folder from April 15th, and it's called Meiosis Stages Blank. So, as you might recall, meiosis has two divisions. We're going to take one cell called the germ cell, and it's going to go through meiosis 1 to create two cells. And then those two cells are both going to go through meiosis 2 to create 4 different sex cells. Remember that before prophase, we have interphase. So over here, somewhere to the left, we're not going to draw it in, is interphase. And that's when DNA replicates and becomes that X shape that we've talked about so often. The other important thing here about meiosis 1 is that we're going to have homologous pairs. And so those are two chromosomes that have the same type of information, but they may have different versions of that information. So for instance, in this chromosome, you might have the gene for, or on both of them, you'd have the gene for hair color, but mom might have the gene or the allele for black hair color, and dad might have the allele for brown hair color. So it's the same type of information, but they may have different versions, okay? These two chromosomes, when they are together, are called a homologous pair. When a cell has a homologous pair, it is called a diploid cell. And so the cell that goes through meiosis is called the germ cell, and it is a diploid cell. Okay, we're going to have a cell here that's going to have 4 chromosomes. So it's going to have a big one from mom and a little one from mom, and it's going to have a big one from dad and a little one from dad. Okay, so this is a homologous pair and this is a homologous pair. There are 2 homologous pairs for a total of 4 chromosomes. It is a diploid cell. As we know in prophase, right, that nuclear membrane starts to break down. The chromosomes condense, they twist up into that X shape. And then the other important part that's going to happen here is we're going to get crossing over. So our nuclear membrane breaks down and crossing over happens. If you remember, meiosis creates genetic diversity. And one of the ways it does that is through crossing over. Right now, we have a chromosome here from mom with sister chromatids. These 2 sister chromatids are exactly the same, right? It's DNA that has been replicated. But if there's crossover, if we swap part of these arms, then this one will have been from all mom. This one will have been mostly from mom with a little bit from dad. This one will have been a little bit from mom, mostly from dad, and this one all from dad. And so there is none of the sister chromatids are now exactly the same. All right, so we've made it through prophase 1. We're going to head on over into metaphase. I want to make a note here that the chromosomes are going to pull this way, okay? And so when we have our metaphase plate or the middle of the cell, it's going to be kind of down the middle like this, okay? So our homologous pairs are going to line up in the middle, and this is different from my mitosis. Whoops, I got to show that crossing over effect. Right, because the homologous pairs are lining up. Whoops, this is going to have a little bit of orange with blue, and this is going to be all blue. Okay. Right. So they are lined up in the middle of the cell, and during anaphase, these homologous pairs are going to pull apart. If you look closely here, you can see the effect of crossing over, right? So I have a sister chromatid here that's all from mom, mostly from mom, a little bit from dad. Here we have one that's a little bit from mom, mostly from dad. And then over here we have one all from dad. So we've created some genetic variety. So in metaphase, our homologous pairs are going to line up. Then again, if we move over to anaphase, I'm going to make those arrows showing which way the chromosomes are going to go. Here's our middle. And so now those homologous pairs are going to pull apart. So this homologous, or this one from the homologous pair is going to come over here. So we've got a chromosome all from, or a a sister chromatid that's all from mom. Here we have one that's mostly from mom, but a little bit from dad because of that crossing over. Over here, we have one that is a little bit from mom, but mostly from dad. And then we have one all from dad. Okay. So those homologous pairs have split. And then over here, we've got one from dad. And here we've got the one from mom. And so that's anaphase. And the important part about anaphase 1 is that the homologous pairs pull apart. Okay, that is the most important part about anaphase 1. So now these are going to go into a cell, and these are going to go over to a cell, and we're going to show this in telophase. And if you recall, in telophase, the nuclear membrane starts to come back. Okay. And we're going to now have 2 chromosomes in each of these cells. So these 2 chromosomes are going to end up here. So again, I'm going to show the effect of crossing over. So I've got this chromosome and this one. Keep in mind, these 2 here are not homologous pairs. They are not homologous. They are not the same size. They do not have the same centromere location. They don't have the same genes. They are different. This would be like chromosome 1 and this would be chromosome 2. They are not homologous pairs. Over on this side, I have that little chromosome, and then I have the bigger one that has the little bit from the crossing over from mom. Okay? Now, here, are each of these new cells, are they diploid or haploid? Do they have a homologous pair or no homologous pairs? And if these are the homologous pairs, right, we don't have them anymore because we've got a big one and a little one, but we do not have a big one and a big one. We don't have the homologous pairs. They have been split. And so here is where our cells become haploid. And you can also do this by counting chromosomes. So in this cell, there are how many chromosomes? Well, if we count centromeres, we have 1, 2, 3, 4. But over here in this cell, if we count centromeres, we have 1, 2. Okay? So this is a diploid cell. These cells are now haploid. All right, so we're going to head on over into cytokinesis 1, where those cells are going to split. The nuclear membrane is going to come back. The DNA is actually going to unravel and become kind of that chromatin, that kind of tangled spaghetti mess, but we're not going to show that. So I've got my big one and my little one that has my little tip from dad. And then I've got my big one and my little one that has the little tip from mom. Okay. These now have 2 chromosomes each. And I want you to note and take a look. Are these chromosomes replicated or are they unreplicated? Right. Well, they're replicated, right? They have a copy of their DNA. And so what's going to happen during meiosis 2 down here? Well, those sister chromatids are going to pull apart. And this is very, very, very similar to mitosis, which you know pretty well. All right, so we're going to head down into meiosis 2. And here we are at prophase 2. And this is a lot less eventful than it was in prophase 1. There's no crossing over, but that nuclear membrane does break down, and then those chromosomes recondense. And so again, we have the big one and the little one with the little tip from mom. Oops. Make that a little clearer. Right, and then down here, we have the big one and the little one, like chromosome 1 and 2. But again, we have that little tip from dad because of the crossing over last time. So that nuclear membrane breaks down, the chromosomes condense, and then we're heading over to metaphase 2. And again, the chromosomes are going to pull apart this way. Now, why are there 2 cells here, right? Up here we only had 1, but down here we have 2. Well, because these cells came from cytokinesis 1. So we have 2 cells here, and those 2 cells are now going into prophase 2. All right, are these cells identical? Well, no, right? Because these might have different versions of the genes. So this one might have the allele for black hair and green eyes, and this one might have the allele for brown hair and blue eyes. And so while they have the information for eye color and hair color, it's different versions. So there is some genetic diversity here. All right, now we're in metaphase 2. Here's the middle of our cell. And so now each one is going to separate. So what's going to line up in the middle? Well, it's not homologous pairs like up here. It's just the regular chromosomes that are going to line up in the middle. So we have the big one with a little bit of orange, and then we have the little one, okay? And then down here, we've got the big one with a little bit of blue, and then we have the little one, oops. Okay. And now we have those chromosomes that are lined up in the middle during metaphase 2. What's going to happen during anaphase? Well, what's going to pull apart? This is important. The sister chromatids pull apart. I want you to, star, circle, highlight, underline these. These are very, very, very important. In meiosis 1, the homologous pairs pull apart. In meiosis 2, the sister chromatids pull apart. All right, so in each cell, we've got our chromosomes in the middle, and now they're going to pull apart. Those spindle fibers, which I'm not drawing to make it not confusing, but are shown in a lot of the videos, those spindle fibers are going to pull those chromosomes apart, those sister chromatids. All right, so on this side, we're going to have, when this one splits, the one that's going to be going this way, again, we're pulling apart this way. Okay. This one is going to be this sister chromatid. So it's all blue with a little bit of orange. And over here, this one's all blue. Okay. That's that big chromosome. Then we have that little chromosome. Right. Down here, similar thing, just the reverse. We have a big orange one and a little orange one, but this one happens to have a little bit of blue because of that crossing over. Over here we have a big one that's all orange and a little one that's all orange. Then in telophase 2, those nuclear membranes are going to come back. Okay. And you can start to see where we're going to get 4 individual cells. And in this one, it's going to be the chromosomes from here. So we're going to have a big blue one and a little blue one. Here we're going to have a little blue one and a big blue one, but there's going to be some genetic diversity because that one has a little bit of orange from that crossing over in prophase 1. Down here, we're going to have one that's all orange, a big one and a little one. And then down here, we're going to have a big one and a little one that are both all orange, with the exception of that little bit of blue from that crossing over. Then if we take these and put them into cytokinesis 2, now we have 4 sex cells. In males, these will be sperm. In females, these will be eggs. It's a little more complicated, but that's what we're going to go with for now. And I'm just going to straighten out these V's into sticks to make it a little easier to see. So now we have, sorry, there's terrible shadow there, a big blue one and a little blue one, right? And then we've got a little blue one and a big blue one that has a little bit of orange from the crossing over. And then down here, we've got a big one that's orange and a little one that's orange. And then we have a big one that's orange and a little one that's orange, but this one has a little bit of blue from that crossing over. So now we have these sex cells. And if you look, are any of them the same? Well, these ones have 2 blue ones, but this one is similar, but it has that little bit of orange. And this one has all orange, and this one is similar, but it has a little bit of blue. So each one of these is unique. They are unique. That is one of the things about meiosis and sexual reproduction, is that you get unique cells. And are these haploid or diploid? Well, let's look. We started out with how many chromosomes? 1, 2, 3, 4. If we go down here, how many are in each cell? 1, 2, 1, 2, 1, 2, 1, 2. So these are haploid. Okay. And how are they? These also here were also haploid, but how are they different? Well, these have only one of the sister chromatids, where these have replicated chromosomes. And so these are ready to go because their sister chromatids have been split. So these are haploid sex cells, right? And they are each unique. Now we've shown this just with 2, but we know that we have 46 chromosomes or 23 pairs. And so if you have all this crossing over happening with 46 different chromosomes, you're going to get a lot of diversity. You're going to get a lot of differences. There's a second part that creates genetic diversity that I'm just going to touch on really, really, really quick. And it's how the homologous pairs line up. So in this case, we have both the ones from dad on one side and we have both the ones from mom on one side, right? So that has created sex cells that look like this. But what if these lined up so that the orange one was on this side? So we had a big blue one and a little orange one, and a big orange one and a little blue one. Can that happen? And the answer is yes. That's called independent assortment. Okay, and so these all line up independently. It doesn't always line up with all blue or all orange. There's always a mixture. So when you add independent assortment along with crossing over, you get a tremendous amount of genetic diversity. And so then no matter how many eggs or sperm you make, all of them are unique. And then when they meet with your partner's egg or sperm, if you decide to have children, you get a tremendous amount of variety. I hope that's helpful. If you have questions, please shoot me an email or your biology teacher an email. I hope you're doing all well. Take care, and we'll talk to you soon.